Hi, I'm Danilo Dubocanin, a computational biologist in the Stergakis lab at the University of Washington Department of Medical Genetics, and I'm excited to present to you a novel statistical framework for single-molecule transcription factor footprinting. Our framework to detect transcription factor footprinting on single molecules of chromatin is built upon FibreSeq. So what is FibreSeq and how does FibreSeq work? FibreSeq leverages an exogenous M6A methyltransferase to stencil in the underlying chromatin architecture of a fiber. We couple this with PacBio long-read hi-fi sequencing to generate highly accurate fibers, approximately 20 kilobases in length. And we also use the PacBio polymerase kinetics to discern M6As along each individual fiber. To confidently determine the presence of an M6A, we apply a Gaussian mixture model to the kinetic information obtained. This allows us to have highly accurate genetic information and also understand the underlying chromatin architecture of an individual fiber across multiple kilobases. We can break down the workflow of transcription factor footprinting into two problems: the de novo problem, where we scan the entirety of the genome for footprints, or the informed problem, where we look at predefined motifs or loci for the evidence of a footprint. Both problems require that we control for the intrinsic methylation preferences of the M6A methyltransferase, and to do so we generate a background model with methylation probabilities for each 7-mer from a deprotonated control sample. For both problems, we must also determine what regions of the genome are accessible to transcription factors. And to do so, we apply a hidden Markov model on each fiber to find the nucleosomes based off our M6A data. We deemed that the non-nucleosomal regions larger than 40 base pairs are accessible to transcription factors for binding. In the informed footprinting approach, we score each provided loci along each fiber based on the methyltransferase methylation preference. In the de novo approach, we score each region with 3 or more non-methylated adenines based on methyltransferase methylation preferences. For both workflows, we then apply a methylation density and footprint size specific false discovery rate threshold to discern highly probable footprints. We performed our analysis on the GM12878 cell line. Being able to accurately discern nucleosomes is crucial in correctly detecting transcription factor footprints. The majority of the nucleosome-free regions that are accessible to our methyltransferase are under 115 base pairs, and these shorter regions, less than 40 or 50 base pairs, represent the internucleosomal region between nucleosomes. On the right side, you're seeing a graph that represents the distribution of nucleosome sizes, and this bimodal distribution represents both single nucleosomes as well as dinucleosomes. The peak in this graph corresponds to 149 base pairs, which is consistent with the crystal structure size of a nucleosome. The bottom graph represents the aggregate M6A profile along fibers. We observe an oscillatory pattern between methylated and unmethylated regions, representing the spacing observed between nucleosomes. We applied our informed footprinting approach to interrogate CTCF binding in GM12878 cells. To generate high-quality potential CTCF binding sites, we combined FIMO-generated CTCF motifs, DNase-1 CTCF footprints, and CTCF ChIP-seq data. The top panel represents the aggregate binding profile from 3 populations of fibers: purple representing CTCF-bound fibers, blue representing accessible but unbound fibers, and orange, inaccessible and thus unbound fibers. Each line in the bottom panel represents a single chromatin fiber. The color of the fiber denotes the population it falls into. The defining feature of the bound population is the large stretch devoid of methylation in the M6A-rich region. This can be observed in the aggregate data, as well as along each individual chromatin fiber. The unbound, accessible fibers have high levels of M6A surrounding the CTCF motif, but within the motif, the relative M6A level is representative of the intrinsic methylation preference. To visualize our de novo footprinting approach, we looked at all the potential binding sites of 2 transcription factors, SOX3 and MYF6. There is a clear drop in M6A signal where the SOX3 or MYF6 binding motifs are in bound elements, and as before, in unbound and accessible elements, the M6A frequency is representative of the intrinsic methylation preference of the methyltransferase. With this approach, you can also directly quantify the number of motifs or binding sites that are bound by a specific TF. In this case, there is more SOX3 binding in these GM12878 cells than there is MYF6 binding. Looking forward, we are excited to use this tool to probe the regulatory dynamics of transcription factors along single chromatin fibers. I'd like to thank my PI, Andrew Sturgakis, and for his mentorship, and also Dr. Daniela Witten for assisting in developing our statistical methods for this problem. I'd also like to thank everyone else in the Sturgakis lab for their support.